The University of Michigan Division of Gastroenterology and the Inflammatory Bowel Disease Program host the IBD Visiting Professor Lecture Series in Ann Arbor, Michigan. This series presents the latest in clinical research and basic science from IBD experts from around the world.
This is believe it or not, is Cleveland. This is downtown. This is Lake Erie. So going in that direction, you end up in Ann Arbor. And this is the campus of the Cleveland Clinic, which is pretty large and impressive, and that's where I spent many, many years of my life.
Point out your hotel, Claudio. Point out where the hotel is.
The hotel?
Very impressive.
The hotel that you stayed is behind the tower.
That's what we need to do.
So, I hope that the title doesn't turn you off too much. But I actually copied the first sentence from a paper that you may have seen. Hello? In Science, not too long ago, where they did the list of the 10 most important developments in the last decade, science-wise. And as I was looking at this list of what smart people picked, I realized that there were 4 crucial components that are probably relevant to IBD. First of all, they talk about DNA. Then they talk about the new concept of cell plasticity. Cells are not really terminally differentiated. They probably revert into whatever is needed by the body. And then they talk about inflammation, as obviously related to IBD. And then they talk the hard-working gut microbes. So these are basically 4 components that now are pillars of inflammatory bowel disease. So I think that people that do research in IBD, they are in good shape, or well positioned, because if somebody like science say this is an important subject and we're all doing them, I think that we are probably on the right path to find out what is IBD all about and how to manage it. Then I want to go back in time and show you a slide that I developed in 1998. Looking back, IBD a little over a decade ago, and if we look at what we knew, what we thought were the components of IBD back in the late 90s, the genes, the environment, bacteria, and the mechanism leading to inflammation, I think we were already on target as far as knowing what are the key elements. And in fact, these key components of IBD pathogenesis are still the one they will focus on, except that now we have picked particular areas and particular group of individuals go into each one of them. And as you start to do that, obviously things become more complicated, because each one of them is an enigma in itself. So what we have a variety of components of IBD, which go from endogenous to exogenous factors, and all of them will act probably in a very coordinated but still unknown way and cause IBD in somebody. So this is the results of very complex pathways. And the complexity, which is in my title, also do with the fact that we have many components and we still don't understand how they fit all together. So we have a complexity of IBD pathogenesis, but we still have the patient, and each patient with IBD is somewhat unique. They have different location of the disease, different response to therapy, different symptomatology, and so on. So I think with what we're facing now, we know probably what causes the disease, not the mechanism necessarily, and we have the response in the patient, but we don't know how we have so many factors and so many individual responses. And starting through the pathways of looking at the factors, if we look at the component of IBD genetics, in 1932, Joe Kirshner, Burley Cron defined terminal ileitis. It took 30 years for Joe Kirshner to say, well, actually, we have these patients in families. We have multiplicity of patients in the same family, suggesting environmental, but more so genetic factors. Then it took about another only a little time to get in, 30 years to get to the genome scan. And then we found the first gene in 2001, the NOT2. And then it took only 5 years to find the second gene, the IL-2 receptor, the IL-23 receptor. And then now, in only a few years, we have more than 100 genes associated with inflammatory bowel disease. And what we're doing now is keep accumulating long list of genes, loci, risk factors, variations that are associated with inflammatory bowel disease. And one of the questions is, how many more genes do we have? What we know now explains about 30% of genetic background. So there's a lot of people that have genetic risk factors that are very rare, and they are associated with very small group of people. But this represents still the majority. And there's a major challenge in now understanding how the genetic variations relate to inflammatory bowel disease. And if you look at the NOT2 that has been around 10 years, we still don't know how NOT2 causes terminal ileitis. And one way possibly to approach this is to look at genes not individually, but all together, and ask the question, are genes whose variation is being associated with IBD in similar pathway, so that perhaps they belong to complex integrative pathways that lead to the disease? It's very unlikely that one single variation in a single gene does that. And this diagram by Akanarson at CHOP proposes an example like this, in which there is variations of the L2L pathway that are associated probably in the same cells, and the combination of genetic variations that maybe can lead to major changes in the behavior of the immune response. And this is true for all the other types of genes. They span different categories of elements. And from this somewhat sophisticated approach of GWAS, genomics, now we start to see reports that are not particularly impressive, but in which we can see the trends of the IBD community in trying to integrate big chunks of knowledge into expression profiles or genetic signatures. These are not big studies, but nevertheless, there's a hint that by studying responses in more global fashion, we can now perhaps categorize patients into patients that will respond to infliximab or fail to respond, predict surgery, predict outcome, and so on. So again, I think we are in the right trends. We just need to become more sophisticated in the analysis of genetic factors. What I personally think that is by far the most important components of IBD is the epidemiology of IBD. What you see here from the dark pink to the light pink is the variation over time of incidence and prevalence of IBD in different countries. And from what was basically North American, North European disease right after World War II, now we see IBD everywhere in the world, and the last people to suffer are in the Asian continent. So this is, to me, is a fundamental piece of information that tells us that as we evolve as human beings, we develop westernized, more, quote, civilized way of living. Whatever we do to ourselves predispose us to IBD, and this could be reproduced for MS, psoriasis, and many other chronic autoimmune diseases. So in many ways, I think that we make these diseases for ourselves. And what do we do to cause disease? What have we changed in the environment? Well, people forget that what's in the gastrointestinal tract is two things only, basically: bacteria and food. And when we ask the question, we always say, well, it's bad to have hamburgers and greasy food and a lot of sugars and so on. But we don't understand how this impact. And if we ask the question, are food-derived components involved in IBD pathogenesis, intuitively one says, yeah, probably more likely, because the association in time and space is there. But when we ask the question specifically, how does that happen, we have no clues. Nevertheless, we are arriving to a point where we understand that what we're exposed to early in life, very early in life, probably has a very big impact on the development of disease later on. This is a nice study from the New England Journal at the end of 2010, showing that depending on the type of milk that certain children drink very early, at age 0 to 10 years, has an impact on the presence of autoantibodies, in this particular case for beta cell autoimmunity. So the chance of developing autoimmunity in life probably is impacted somehow, but how our immune system is educated early in life. And this is fundamentally important, because if we're going to cure the disease in older children or in teenagers, in adults, and the causes are early in life, like in the first few months or the first few years of life, it becomes fundamentally important to change lifestyles, we understand what we do wrong early in the game.